Welcome to Stanford MedStory Podcast. I'm Katie Kanagawa, Director of Communications for Stanford Department of Medicine.
And I'm Rebecca Handler, Manager of Scientific Communication Strategy. Today, we're discussing research from our own Stanford Medicine team that could fundamentally change how we approach vaccine development, not just for malaria, but potentially for many diseases.
That's right, Rebecca. This collaboration between Stanford researchers and Ugandan scientists has revealed something unexpected about how children's immune systems learn to manage repeated malaria infections, and it challenges everything we thought we knew about protective immunity.
Katie, let's start with the scope of this challenge. In parts of Uganda, children experience malaria infections repeatedly, some up to 30 times before reaching their teenage years. Yet many of these children don't develop severe disease. Our Stanford team wanted to understand why.
Exactly. The research team, PhD candidate Jason Niduffer, along with Dr. Prasanna Jagannathan and Dr. Maria Grazia Roncarolo, worked closely with Ugandan researchers to study these children's immune responses in unprecedented detail. And what they found was remarkable. For the first time ever, they were able to track individual T-cell clones through multiple malaria infections.
That technical achievement alone is noteworthy. Can you explain why tracking these specific immune cells matters so much?
Of course, T cells are critical components of our adaptive immune system. They remember previous infections and mount targeted responses. By tracking the same T cell families over time, the team could see exactly how these cells were learning and adapting with each malaria exposure. It's like following the educational journey of individual immune cells.
And what they discovered about that journey was surprising, wasn't it?
Absolutely. They found that nearly 90% of the malaria-specific T cells belonged to a previously understudied group called TR1 cells. Now, for our listeners, these aren't the typical fighter cells we usually hear about. TR1 cells are regulatory cells. They manage the immune response rather than directly attacking pathogens.
This seems counterintuitive. Why would the immune system develop cells that regulate rather than eliminate the infection?
This gets to a crucial insight about malaria pathology. The severe symptoms of malaria, like cerebral complications, severe anemia, respiratory distress, these are actually caused primarily by the body's own inflammatory response, not directly by the parasite itself. When our immune system overreacts, that inflammatory cascade can be deadly.
So these TR1 cells are preventing that overreaction?
Precisely. They're maintaining a controlled response that keeps the parasite in check without triggering dangerous inflammation.
So let's talk about the implications for vaccine development. Traditionally, we've aimed for vaccines that prevent infection entirely, what's called sterilizing immunity. But this research suggests a different approach might be more effective.
That's the paradigm shift here. Instead of trying to prevent all infections, we might develop vaccines that train the immune system to manage infections without severe disease. The idea is that controlled infections could actually boost immunity over time, while the TR1 cells prevent severe illness.
Let's talk about the methodology for a moment, because this was a remarkable collaborative effort. How did the Stanford-Uganda partnership make this discovery possible?
The collaboration was essential. Our Ugandan colleagues provided not just blood samples, but crucial epidemiological context, understanding infection patterns, seasonal variations, and clinical outcomes in these communities. They've been observing these phenomena clinically for years. What Stanford brought was advanced immunological analysis, single-cell RNA sequencing, T-cell receptor sequencing, and computational approaches to track clonal lineages.
It's worth emphasizing that this level of detailed immune monitoring has never been achieved before in human infection studies. The team could literally follow the same immune cell families as they encountered malaria again and again.
And that longitudinal view revealed how these TR1 cells were consistently selected and expanded with each infection. It's natural selection happening within the immune system, favoring cells that promote survival through regulation rather than aggressive elimination.
Now, Katie, how might this translate into real-world applications? What are the next steps?
The team is already working to identify specific antigens or markers that can help boost the growth of TR1 cells. If we can incorporate these into vaccines, we might be able to train immune systems to develop this protective regulatory response before children experience their first severe malaria infection.
And the implications extend beyond malaria.
Absolutely. Think about COVID-19, where we saw that severe disease often resulted from an overactive immune response or autoimmune conditions where the immune system lacks proper regulation. This TR1 focused approach could inform treatment strategies for many conditions where immune overreaction is part of the problem.
There's also an interesting global health equity angle here. This discovery was only possible because of long-term studies with children in Uganda who live with repeated malaria exposure. Their experiences gave insights into immune responses that we could never see in low exposure settings.
That's such an important point about learning from the remarkable adaptations that have evolved in these populations. Ugandan children's immune systems have developed an elegant solution to a deadly challenge, and now researchers are working to understand and potentially replicate that solution through vaccination.
Let's address what might seem like a concerning aspect to some listeners, the idea of a vaccine that allows infection. How do we think about safety in this context?
It's a valid concern that requires careful consideration. The key is that we're talking about controlled, manageable infections, not severe disease. The data from these Ugandan children shows that with proper TR1 responses, repeated infections don't lead to severe outcomes. In fact, they may contribute to building a robust, long-lasting immune response conducive to infections that are totally asymptomatic.
And this isn't theoretical. We're seeing it work in real-world conditions among these children.
Exactly. Despite continuous exposure to malaria, these children are still going to school, playing, living normal lives. Their immune systems have found a sustainable balance, and that's what we're hoping to replicate through vaccination.
The research team also noted that this discovery required new computational approaches to handle the complexity of tracking immune cells over time. Can you speak to the technical innovation involved?
The computational challenges were significant. The team applied advanced computational methods to track T-cell receptor sequences across multiple time points and infections. Each T-cell has a unique receptor, like a molecular fingerprint, and following these through the noise of billions of other immune cells required sophisticated bioinformatics approaches that the Stanford team leaned on for this study.
It really showcases how modern immunology requires this intersection of wet lab science, computational biology, and field epidemiology.
Absolutely. And none of it would be possible without the trust and participation of the communities in Uganda who've been part of these longitudinal studies, some for many years.
This research from the Stanford-Uganda collaboration really exemplifies how breakthrough discoveries often come from questioning fundamental assumptions. In this case, the assumption that vaccines must prevent infection to be protective.
Indeed. The work by Jason Nidafer, Prasanna Jagannathan, Maria Grazia Roncarolo, and their Ugandan colleagues has opened an entirely new avenue for vaccine development, not just for malaria, which affects millions of children worldwide, but potentially for other diseases where immune overreaction contributes to pathology.
For our listeners who want to dive deeper into the science, the full study is available in Science Immunology, and we'll include links in our show notes. You can also find more information about Stanford Department of Medicine's global health initiatives on our website, medicine.stanford.edu.
Thank you for joining us for this Stanford MedStory podcast. I'm Katie Kanagawa.
And I'm Rebecca Handler. We'll be back with another breakthrough discovery from Stanford Medicine that's advancing human health.